Good afternoon, my name is Ekaterina Mironova. I am PhD, head of the Laboratory of Molecular Neuroimmunoendocrinology, St. Petersburg Research Institute of Physiopulmonology, and also senior researcher of Laboratory of Molecular Mechanisms of Aging, St. Petersburg Institute of Bioregulation and Gerontology. Today our topic is application of peptide bioregulators for the prevention of retinal diseases. The problem of retinal diseases is one of the most pressing in ophthalmology. Such diseases as central artery obstruction, retinitis pigmentosa, diabetes retinopathy, retinal detachment, and rupture often cause blindness. According to the World Health Organization, the 21st century retinal pathology, along with the cancer, is becoming one of the leading cases of disability in the world. The retina is a thin nerve tissue located on the back wall of the eyeball. It contains millions of light-sensitive photoreceptor cells that convert light signals into electrical signals. These signals are then transmitted via the optic nerve to the brain, where they are interpreted. In most cases, at an early stage of retinopathy, the disease doesn't manifest itself in any way. That is why the pathology is often diagnosed when the pathological process is already progressing. Currently, modern methods of treating retinal pathology include pharmacotherapy, laser treatment, and surgical treatment. Pharmacotherapy is varied but ineffective. In some countries, hemostatic drugs, blood-improving drugs, glucocorticoid drugs, various biostimulants, and adaptogens are used to reduce the risk of new eye complications. The main goal of conservative therapy is to influence the pathogenesis of retinal pathology. This explains the use of drugs that strengthen the vascular wall, angioprotectors, improve retinal trophism, adaptogens, block free radicals that occur when oxidation reduction processes are disrupted, antioxidants, etc. However, conservative therapy is effective only in the early stages of the processes. Its results are unstable. It usually helps to stop or slow down further vision loss, but doesn't improve it. Therefore, this treatment method is used only as a maintenance therapy. Scientists from the St. Petersburg Institute of Bioregulation and Gerontology have discovered an effective combined use of visoluten, eye tissue peptides, serluten, brain peptides, and Wentworth, vascular peptides and retinal pathology. This combination of drugs helps to normalize visual function in patients by regulating apoptosis, proliferation, and differentiation of retinal neurons, and also helps to maintain and improve microcirculation in the retinal vesicles. Visoluten application in patients with dystrophic retinal diseases in the main group of examined patients increased visual acuity in 58% of cases, while in the comparison group, use of conventional means in 26% of cases. Expansion of the peripheral boundaries of the visual field in the main group of patients was observed in 64% of cases, and in comparison group in 40% of cases. A decrease in the total area of absolute stochomas in the main group was noted in 42% of cases, and in the comparison group only in 20% of cases. Thus, in retinal diseases, visoluten improves visual function and promotes increased visual acuity expansion of the peripheral boundaries of the visual field and a decrease in the total area of absolute scotomas after the first courses of application, and also has a positive effect of the preservation of visual function with regular long-term application. Based on the analysis of the amino acid composition of the Wentworth polypeptide complex isolated from the vessels of young animals, the Vasogen 3 peptide with amino acid sequences KED was synthesized. In organotypic cell cultures obtained from young and old animals and in dissociated cultures of vascular endothelial cells during their aging, Vasogen stimulated the synthesis of the proliferative protein Ki67, the expression of which decreased during cell aging. Vasogen increased the expression of vascular endothelial growth factor in red vascular cultures during their aging. VEGF is produced by cells to stimulate vasculogenesis. This is the process where is the vascular system formation. And angiogenesis, new vessels grows in an exciting vascular system. Based on the analysis of the amino acid composition of the cerulin polypeptide complex, which was isolated from the brain tissue of young animals, the 3-peptide pinealone amino acid sequence EDR and the tetrapeptide IEDP was synthesized. In vitro studies have shown that the EDR peptide regulates gene expression and protein synthesis responsible for the functional activity of neurons. It has been established that the EDR peptide affects cellular processes such as apoptosis, proliferation, and neurogenesis, the transformation of cell into mature neurons. In addition, it has antioxidant properties. Bioinformatics methods have confirmed that the mechanism of gene expression, regulation, and neurogenesis protein synthesis under the influence of the EDR peptide may associated with its ability to interact with the DNA sequence, which you can see on this picture, in the classical bed form. It has been shown that the peptide ADP has a wide range of neurotrophic effects. Experimental data indicate accelerated and complex restoration of functional and structural characteristics of regenerating peripheral nerve sensory axons after its mechanical compression under the IADP peptide influence. Thus, the St. Petersburg Institute of Bioregulation and Gerontology has developed a unique technology for restoring the damaged retina in various diseases. The technology is based on the complex application of peptide bioregulators isolated from the retina, blood vessels, and brain, as well as their synthetic analogs. The combined application of Visaluten, Serlutin, and Wentworth allows for a beneficial effect of all links of the pathological process, from restoring the functional activity of retinal neurons to maintaining and improving microcirculation in its vessels. The results of the widespread application of bioregulators contribute to an increase in the period of working capacity, a decrease in the disability percentage, and an improvement in the quality of people's life, which can have a positive effect on the social and economics indicators for the population medical care. Thank you for your attention.